So here you see a diagram of the entire process of meiosis. Now remember, meiosis is the process of making four gametes, or four sex cells, that only have half the genetic information that's necessary for an organism to survive. So in picture A, we're going to see a cell that is in interphase. We can see a nucleus intact. This might be getting towards prophase because we can see that all of our chromosomes have paired up, and we see identical copies of each of the chromosomes. So one half of this X was mom's chromosome and the other half is the copy. And then this is the chromosome from dad and its copy. At this point, we're starting to see that the spindles are showing up and the nucleus is starting to fade away. So this is probably closer to prophase 1. During prophase 1, we also have what we see as crossing over. So some of the information from the chromosome and its copy that came from mom is being swapped with the chromosome and its copy that came from the dad originally. So in the long run, we're actually talking about grandparents' genetic information being swapped. So after prophase 1, we're going to go to metaphase 1. And in metaphase 1, those chromosomes, the one from mom and its copy and the one from dad and its copy, are called homologous chromosomes. Remember, the prefix homo means same, so they're coding for the same traits. It's just the traits from mom or the traits that come from dad. So those homologous pairs are lined up down the center of our cell. Notice that they're 2 by 2. So they're not in a single file line, they're doubled up as a homologous pair. So during anaphase 1, one half of that homologous pair gets split from the other half. So this means like chromosome 1 and its identical copy go to one side, and chromosome 1 and its identical copy from dad go to the other side. Now remember, I'm saying mom and dad, but now because crossing over happened in prophase, there's a mix of genetic information. Then we move on to telophase 1. In telophase 1, we get two nuclei. Notice that this has chromosome 1 from maybe mom and chromosome 2 from dad, chromosome 1 from dad and 2 from mom. So this is telophase 1. After telophase 1, we continue to have cytokinesis occur. So this cell completely breaks, and now we have two cells. These only have half the genetic information that's necessary, but they have half in its copy of everything. So it kind of looks like we have enough, but we don't really, because remember, it took two copies and its pairs to make that original cell. So now we've gone on, and this is prophase 2. During prophase 2, we've got these, what's left of our homologous pairs, one on each side. And then we start to have the spindles reappear, the nucleus starts to break down, and we go into metaphase 2. The second time we have metaphase, there are no homologous pairs anymore. They're in two separate cells. So we're just going to have these chromosomes line up in one single file line. So in metaphase 1, they're 2 by 2. In metaphase 2, they're all in a single file line. So these are sister chromatids, the left and the right. Just like we had in mitosis, the only difference is because of crossing over, they're not perfectly identical to each other like they were in mitosis. There's the mixing of genetic information on each of these chromatids. So just like always, our spindles are going to attach at the centromere. They're going to, during anaphase 2, separate those chromatids. The chromatids are going to move to opposite ends of the cell, and we're going to go through telophase 2, where two new nuclei are forming in each of those cells. So now we have 4 nuclei in 2 cells. This will continue to go through cytokinesis, and we'll be left with 4 separate cells that each now have half the genetic information as the original parent, and each one is genetically unique because we have only half the genetic information, but crossing over happens, so we have swapping of each half. So we're going to get a wide variety in each one of these daughter cells. This leads to the genetic variety that we're going to see in the offspring. So genetic variety in the offspring can be because of what happens with crossing over, depending on what order those homologous pairs line up in, and then what order the sister chromatids, meaning left or right side, that genetic information ends up on. So we end up with very different and unique daughter cells each time meiosis occurs. This is why the offspring are each unique and different.